Okay, I hope you can all see my screen.
We
Thank
can.
you for the nice introduction. So I'm Tom Mulvaney, and I'll be sharing this presentation today with Aaron Sweeney, and we are both postdocs in Meyer Topf's lab at CSSB here in Hamburg, Germany. And I'm going to be talking about two tools that have come out of the lab recently, TEMPy-ReFF and ChemEM. And the kind of methods we develop in the lab are what we call integrative methods. So the idea is that you take information from different sources. They could be from different experimental techniques like CryoEM or cross-linking data. And we also try and combine those with computational approaches. And TEMPy-ReFF and ChemEM are both tools aimed at modeling using CryoEM data. Since 2014, there's been this resolution revolution. And what that means is that today, the majority of the structures which are being deposited in the PDB and the reconstructions in the EMDB are at better than 4 angstroms in resolution. Of course, this is just a average resolution. So that means that parts of the models will be at much lower resolutions, and parts might even be at higher resolutions. With this resolution revolution, we can also see that CryoEM has become the sort of favored technique now for solving nucleic acid structures. So we can see that today, in fact, the majority of depositions of nucleic acid containing molecules are coming from CryoEM. How we actually go about building atomic models into CryoEM reconstructions is very much resolution dependent. So if we're at low resolution, say 20 angstroms, it might really only be possible to adjust the position of protomers in complexes. As the resolution improves, say to 15 angstroms, it might even be possible to adjust domains within these proteins. And at the sort of intermediate resolution range, so better than 10 angstroms, we can apply what's often called flexible fitting. So the idea here is that we treat the protein or the nucleic acid as something flexible, and we can actually adjust the positions of the alpha helices, the beta sheets, maybe even the residues at higher resolutions. And ChemEM and TEMPy-ReFF, two tools which we'll be talking about, are really aimed at this kind of intermediate to high resolution regime. In the past, flexible fitting approaches would typically rely on models, say, from crystallography or from other cryo-EM experiments. But today, we're in this very lucky situation where we have high accuracy structure prediction methods such as AlphaFold. But still, AlphaFold models are not always perfect, and they pose challenges for us. So here we've got a very extreme example. It's from a pore-forming protein, which was recently solved by Gwen Marini in the lab. And AlphaFold does a really good job of predicting the pre-pore conformation of the protein. But the post pore conformation is not well predicted at all. And you can see here that the pore conformation requires a really large rearrangement of some of the domains, and also a complete change in secondary structure with this formation of a beta hairpin. So in order to model this, Gwen used a combination of flexible fitting, which we'll be talking about later, as well as manual model building in Chimera. There are two very standard methods for performing flexible fitting, and I can break them down into molecular dynamics flexible fitting, or MDFF. And if you're familiar with tools like iSolder, then you've used this kind of method. The other approach is the correlation-based fitting, where you try and optimize the cross-correlation between a model and a map. And this kind of approach was pioneered by Topf and also Florence Tarmer, for example. TEMPy-ReFF is another approach entirely. It's kind of more similar to the correlation method, but we use a Gaussian mixture model. I'm going to explain now why that's advantageous. So the idea with MTEFF is if you've got some experimental density, which is the result of, let's say, two atoms in this really simple case, and we have an initial model, and we want to adjust the position of the atoms in this initial model to match the experimental data. In MTEFF, we push the atoms towards regions of high intensity. So you can imagine the force pushing these atoms. And in this case, we're actually going to be pushing both of the atoms to this peak. And of course, you're going to get a clash then. Now, in practice, the clashes usually don't happen because you have other additional restraints, right? This bond is usually explicitly modeled, so that stops this, but it's still a limitation of this kind of potential. In the correlation-driven approach, we treat each of the atoms as Gaussians, but it's a very simplistic approximation because the width of the Gaussians is fixed, and this is fixed based on the average resolution of the CryoEM map, say. And of course, this is an approximation because CryoEM reconstructions are not homogeneous, right? There are parts with high resolution and parts with low resolution. And the idea is that we adjust the position of these Gaussians and thus the sort of positions of our atoms in our model such that they correlate well with the experimental data. And the GMM approach in TEMPy-ReFF is essentially the same, but these Gaussians have variable widths. So that means we adjust the position of the Gaussians, but also their widths. And by doing so, we're approximating the local resolution differences throughout the map. So TEMPy-ReFF stands for TEMPy responsible flexible fitting, and responsibility is an idea which comes out of Gaussian mixture modeling. I'm not going to explain it in too much detail, but it basically means that the Gaussians are responsible for different parts of the signal. And of course, by adjusting the positions of the Gaussian, we refine the atomic coordinates, but we can also determine the B factors or local resolution. And another downstream step one can do with TEMPy-ReFF is to actually generate an ensemble of models, which can sometimes better explain the experimental data. And I'll look at that a bit later. Unfortunately, TEMPy-ReFF, just like MTEFF and correlation-driven approaches, has a limitation, and that is that it's quite easy for it to get stuck into local minima. And I'm going to try and visually show you this with this little movie. Hopefully it plays. So here we've got a human processome in the post-A1 conformation. That's the reconstruction. And our starting model's in the pre-A1 conformation. And you can see during the fitting process, I'm going to zoom in now, that the structure actually ends up getting a bit distorted during the fitting in this region here because it was so far from the initial, so far from the correct position, it gets pulled into the wrong bits of density. So to get around this, we use restraints. And one of the restraints that we use in TEMPy is rib find. And the idea with rib find is that you break your structure down into rigid bodies, in this case, in a hierarchical fashion. So you break things down into smaller and smaller rigid bodies, and you optimize the position of these. And you can see now, again, if we zoom in, that in this upper region of this processome, that this rib find based approach ends up producing a much better refined model. And you can see that it looks much similar to the deposited model here in pink. So we describe rib find in our paper in nucleic acid research. Rib find 2 is really aimed at breaking nucleic acids down into rigid bodies. So the idea is that you identify secondary structure elements. So in this little panel up the top here, I've got a sort of protein described as a graph where you've got alpha helices and beta sheets. and these edges connecting them, and you remove the weak edges to create smaller and smaller subsets of rigid bodies. And what we saw in this video just now is that using the rib fine restraints, we get a better fit to the density. We could see this visually. But this plot here shows you exactly this using the cross correlation coefficients. You can see in blue, the cross correlation is much higher for the hierarchical approach. And for the unrestrained approach in green, we have a lower cross correlation coefficient. And in the past, before TEMPy-ReFF was around and we used ChemEM, it was very common to use just a single rigid body rather than this hierarchical approach. And you can see that in orange, what we label as UCN here. So TEMPy-ReFF is available in CCPM in the new Doppio release. It comes in a few different flavors there. There's this FlexFit GUI. And this FlexFit GUI is designed to set up a flexible fitting run using the rib fine restraints. And there's also another GUI for doing the GMM-based refinement. And the really nice thing about this integration into CCPM Doppio is that all these downstream tasks which are available become very easy and accessible. So you can use this GMM refinement, or you could use RefMac or ServalCat to further refine your flexible fitting process, or you could even use something outside of Doppio, such as Phoenix. I've got here some results from a benchmark of TEMPy-ReFF. So we compared TEMPy-ReFF refined models against models from Ceres, which is a public repository of Phoenix refined models. And we can see that the cross correlation for TEMPy-ReFF models is slightly better at lower resolutions than Ceres and the PDB deposited structures. At the same time, the molar probity scores end up being a little bit worse here. But at the higher resolutions, the molar probity scores for TEMPy-ReFF are better. And I assume everyone's familiar with molar probity, but molar probity is a measure of the geometry of proteins and also nucleic acids. So a lower molar probity score indicates better geometry. I also want to quickly talk about the ensemble refinement method in TEMPy-ReFF. On the left here, I've got three maps: the experimental map, a sort of simulated map generated from a single model from TEMPy-ReFF, and a model in orange, which is simulated from an ensemble of TEMPy-ReFF models. And visually, what you can hopefully see is that the orange map looks much more like the experimental map. And if we zoom in and actually look at the ensemble of models, we can see here, for example, there's an arginine which clearly exists in two distinct conformations, which TEMPy-ReFF captures. And of course, there are also parts of the structure which are less dynamic in nature, which you can see on the far right. So these side chains are all very, very similar. We tested TEMPy-ReFF in Cas15. So here we've got a bunch of Cas15 targets. These were mostly multimeric structures, some of them quite large. And what you can see is the predictions often have low fit to the density according to SMOC scores. So SMOC is like a local correlation measure. And when you plot the distribution of SMOC scores for a set of predictions, we can see that they're generally lower. But after refinement using TEMPy-ReFF, we have SMOC scores which are much closer to the SMOC scores of the targets provided by the experimentalists. What was quite exciting in Cas15 was it was also the first time that RNA structure prediction was part of the competition. And here we can see a few different RNA targets. Some of them are natural RNA structures, and some of them are also sort of synthetic constructs. What's interesting here is that after refinement, some of the structures actually had better SMOC scores than the target structure, which is really nice to see. And this is particularly interesting given that many of the predictions actually seemed quite off, at least compared to the protein structures, because RNA's structure prediction is a much newer field. And with that, I'm going to hand over to Aaron to talk about small molecules.
Hey guys, I'm Aaron, a postdoc from Myotop's lab. And I'm going to tell you, about the software we built for fitting small molecules into CryoEM maps called ChemEM. So ChemEM works by using molecular docking and molecular dynamic refinement whilst being aware of the CryoEM data. When we tested it, it was accurate across a range of resolutions from 2.2 to 5.6. Also, it allows you to do multi-ligand fitting if you have more than one ligand in a binding site. You can fit them both in simultaneously, which was nice. So a bit more details on how it works. We developed our own docking score, which was an empirical function with various terms for characterizing the protein-ligand interaction. And when we benchmark this against the ortho.venus scoring function, they both had a similar discriminatory power for identifying a correct small molecule conformation from an incorrect small molecule conformation. We then plug this into our search algorithm, and as you can see on the right, just the docking score alone was fairly accurate across a range of protein ligand complexes, which was encouraging. Next, we now introduce the CryoEM data to the docking function. How we do this in ChemEM is we try and segregate the ligand data from the protein data. So we'll start by identifying voxels within your binding site. And as you can see here in the top right, we have the binding site of all the protein data and the ligand data in there. Once we've identified these, we'll denoise them and segment them using different morphological operations and apply a couple of weak thresholds, and then use this as a mask for the experimental density map. And what you should get out is something that looks like this, which is a nice segment of the ligand map, the density map, which should represent the ligand. We then add this to the ChemEM score using the mutual information scoring function. So the mutual information is a goodness of fit metric, similar in a similar way that cross-correlation is. However, what we found is at lower resolutions, there's no difference between mutual information and cross-correlation, but mutual information was more robust to changes in resolution. So at lower resolution, mutual information performed a bit better, which is why we went with it. We weighted this just to bring it into the same magnitude as our ChemDoc score. This is what we use for the docking phase of ChemEM. Then docking solutions are minimized and passed to the MD protocols. Here we use OpenFF and OpenMM as the driver for our MD, simulated annealing. We use amber force field parameters for the protein. Ligand parameters come from the OpenFF SAGE force field. You can include various implicit solvents, and the CryoEM map is included as a bias. So what you can see here to the left is the output of the docking stage, which is fairly well fit, but can clearly be refined. We then run a few cycles of simulated annealing, and what we get out is a much nicer solution for the final ligand. Here are the results of the benchmark we did in the original paper. So at the top, you can see results for high resolution complexes from 2.2 to under 3 Armstrong resolution. We had 20 complexes, and at the bottom, C and D, are the results for the low resolution benchmark we had, where we had 13 complexes ranging in resolution from 3 to 5.6 Armstrong. For each one of these, we also had a high resolution control, either from CryoEM or from X-ray, so that we could ensure that the actual deposited solutions in the low resolution were reasonable. And what we see is with docking alone, ChemDoc score was able to get correct solution in about 80% of cases for both the high and the low resolution benchmark. And in this bar chart, it's split up by how the quality of these solutions compared to the deposited or the controls. And as you can see, there was only a tiny amount less than 1, but most that we defined as correct were less than 2 or 1.5 Armstrong. Now, when we added in the data from the CryoEM map, we increased not only the number of correct solutions that we got, but also the quality of these solutions for both high and low resolution. We checked the cross-correlation against the map of these solutions compared to the cross-correlation for the deposited, and they were fairly similar for both high and low resolution. However, for the ChemEM solutions, the ligand strain energy in the high resolution benchmark were a lot lower than the deposited solution. For the low resolution, the situation was less unclear, but I guess that's low resolution. Sometimes ChemEM solutions were less strained, sometimes the deposit was less strained. So that's how it works briefly. It's available now on Conda Forge, and you run it by using a configuration file just through terminal. This configuration file has two parts. One is all the data you need to run your protein, your ligand, either as a SMILES or an SDF, the centroid of your binding site, your density map, and the resolution it's at. And the second part is this, which stages are enabled. So you tell ChemEM what it's going to run. There are various things you can do. Pre-processing is just loading up, and it makes a couple of interesting things for you. Then there's these pre-processed split density stage, auto split point, and auto split zone. So this is for when we segment out our density within the binding site. Sometimes you'll get multiple discrete densities being segmented. What we can do is we can split these by disconnected densities and only include for the docking phase the densities either closest to your given point or within a radius of a given point. Sometimes it helps with accuracy. Then fitting, which is the docking, and then dock only if you don't want to use CryoEM data, and post-processing, which is the MD protocols. And it's run through the command line with just the ChemEM and your configuration file. Now for this, I wanted to give an example of how you would actually use it, I guess, in day-to-day basis. So I tried to model the ligands from a deposited, using a deposited protein model and a deposited map in a day to see what I could actually get out. So I chose this human dopamine transporter complex, which is embedded in a membrane at a resolution of 3.19. And I just wanted to see what I could actually do with ChemEM in a day. So in the paper, they say this complex had 2 ligands, CFT and MRS7292. So where do you start? So the first thing I did was run a protocol we have, which is rather handy on ChemEM, called AlphaMask. And this tries to look for discrete densities which are not modeled by your protein. To do this, we treat the geometry of we transform the points of the protein into an alpha shape, an alpha spheres, where small spheres are the protein and intermediate spheres we say are like the SASA or pocket boundaries. So we'll only try and isolate densities from around these places. And the output is, as you can see here, a load of discrete densities which may or may not correspond to different ligands. And I have a video so you can see it slightly better. As you can see, around the outside, there are loads of membrane-looking densities. And within the core, you can see there's 2 discrete density that have been segmented, which look fairly ligand-like. So we started in the core. These are the 2 ligands that are the subject of their investigation. Luckily for me, there was one larger ligand and one larger density and one smaller ligand and a smaller density. So I made 2 configuration files, gave them the centroids of these segments, and it fit it fairly well, I think. Also, another thing that's quite important is when I was running this, if I ran, say, I fit one ligand, and then I wanted to fit the other ligand. During the first fitting, the protein is also, the side chains are allowed to be flexible within the binding site. So to track this in my configuration file, I would have to change my protein and then add the ligand. And you can add the ligands by just creating a directory where you throw them all in. And every time you run a new refinement or fitting, it will load up the ligands, and during both docking phases and MD, all the other ligands that are being fit will know that your ligand is there and they'll be able to interact with it, but your ligand itself won't move from how you fit it. So doing this kind of highlighted the importance of this, the short MD step. The top docking solution is shown here. It kind of looks like a reasonable fit, except for this ring system, which is in the completely wrong configuration. This was picked during the docking phase by RD kit, and this conformation won't change during docking because there's no rotatable bonds. So there's two ways you can get around this. The first is you can give it an SDF file that has the correct conformation, but more often than not, you may not know this. So then you can run the MD refinements, and as you can see, it fixed up the conformation of this, this ring system. And this is the final solution, the ChemEM solution in terracotta next to the deposited solution in green. And it looked pretty good, I thought. So next I move on to membrane modeling, and this is where it got a bit harder. So I started with 3 different length ASR chains, which I assumed would represent the hydrophobic core of the membrane. And then I fit each one of these to individual densities that came out of my alpha mask that looked fairly membrane-like. And what I ended up with was something that looks like this. So it actually wasn't too bad. I think if I had done this properly and had more time, I would have needed to trim some of the size of these ASR chains. But for trying to do it in a day, I thought it was okay. But one thing I learned from this was occasionally when you're fitting a ligand, it can, during minimization stages in docking and during the MD stage, where you relate what you fit in your segment back to your full experimental map, your ligands can end up moving into density that is clearly not meant for them. So there's 2 ways that you can get around this. One is as we mentioned before, the system ligands. If you can fit one of these things, you can add this ligand in, and now the other molecule you're trying to fit won't move into that density. Or the second way is you can fit both simultaneously, which is what I did here. You give, in your config file, each ligand its own centroid point to start from, and both docking and the MD phase will try and fit them both simultaneously. I wouldn't advise doing this with too many ligands, because at the moment in the current version, this is quite slow depending on how many more and more rotatable bonds you add to the system. And finally, I had to model the cholesterols from the membrane. To do this, I fit them into more bulkier looking densities around the membrane, and had a look at some homologous structures similar to how they did in the original study, and ended up fitting them, and they looked fairly well fit. I thought they clearly still need some small refinement, and luckily in ChemEM you can do this. So you see here that I have this cholesterol, the body of which is fit relatively well. This group here is clearly should be pointing towards this density. I could start again and hope that it goes in there, or I can just refine this solution here while holding all of these groups steady so they don't move into any other density or anything. To do this in ChemEM, you add a hold fragment statement to your configuration file. You tell it which ligand it is, L0, with the ligands starting from 0 in order that you've added them to your configuration file, after which, and then list all the atoms you want to hold still. We also give you, as an output in the pre-processing step, some things to make this easier because it's slightly fiddly. The first is a schematic of your molecule with each of the atoms labeled, the atoms and their indices labeled. The next, we break it down into using bricks into what we think is significant fragments, and then give you a file which already has these atom keys. You can just copy and paste them in for which atoms you want to hold steady. Now, after this, I reran the MD refinement step, and the misfit region of the molecule has now moved into density that it should have been in anyway. This clearly still needs some slight refinement. However, it was late when I did this, and I wanted to go to bed. And so in 24 hours, using ChemEM, I managed to make a final model which fairly reasonably represented the deposited model. Obviously, it needs a bit more refinement. However, when I checked the Q-scores with the deposited model, they were fairly similar. You could then take this and pop it into whatever further refinement protocols you have, like Phoenix, whatever, to refine further. And so with that, I'd like to say thank you to everyone in our group and you guys for your time. I hope there was something interesting for you in our talk. And if you have any questions, we'd like to answer them.
All right, thank you both. We do have a couple of questions and we have plenty of time. So if anyone in the audience has more questions, please feel free to submit them. How large can one go with these methods in terms of number of atoms and or number of chains effectively before, the time for these simulations takes too long?
I can't tell you exact numbers, but in general, it's usually not the time that seems to be the problem, but rather like running out of GPU memory. So, I mean, both of our methods rely on OpenMM for doing the MD steps. And yeah, if you have a large system, it will use up a lot of GPU memory. So,
but for ChemEM in particular, you can split this up. I guess if you want to take a smaller, like one subunit of your protein, this will make it faster and allow you to do more. In terms of ligands, it is designed for small molecules. Like something like a really, really large acyl chain should actually be feasible, I guess. Maybe maybe once you get to peptide kind of size, it might take a bit too long. Yeah, but for most small molecules, it will, and it's fairly fast because we only have to fit a small molecule versus refining the whole protein.
Yeah. Yeah, I actually can't tell you what the largest size structure we've refined is. I think in Cas15, some of the largest structures might have had like 3,000 residues. So, these are pretty large molecules, and we managed to refine it on our what have we got? A5000 or something. It's got 24 gigabytes of GPU RAM, and it didn't end up being a problem.
So a follow-up question was, can one use GPUs? And you've clearly answered that. But to flip that around, does the software work on systems without GPUs?
Yeah, ChemEM does. So you can run it. You just need to change the platform which you run the MD on. To be honest, for half of the example I showed, for a lot of it I was at work and I used my machine which had my GPU and my 96 CPUs. Then I got hungry and went home, and I did the rest on just the MacBook.
Okay.
So for small molecules, it's not really a problem. I guess maybe it's different.
Yeah, I mean, TEMPy-ReFF's the same. You can run on a CPU as well, and again, that has the advantage of often you have more RAM on your system than your GPU has, but it will run slower. Yeah.
And to follow up on the size question, is there a limit on the size of ligands one can include?
No, it does tend to, so it's less the actual MD refinement, it's more the docking step search function, which limits the size of ligands. And the limitation is basically on the number of rotatable bonds. Once you start getting up to like 20 plus, or multiple different ligands, it can be a bit slow. But I mean, with like, yeah, it will still run. It just it may be slower. We are working on an updated version, which is a lot quicker, and the number of ligands or size of ligands is no longer an issue. This was done for a couple of months though.
Can one refine the ligand portion and or the protein portion of a complex independently, that is, to apply a constraint to one and just refine the other?
So actually at the moment, this is the biggest limitation with TEMPy-ReFF, is that the parameterization of the molecule only works with proteins, nucleic acids, and water molecules. We have a version which is about to be released, which handles sugars, but ligands at the moment are not handled. So you have to refine those separately using ChemEM.
For ChemEM, it will refine both the ligand and the binding site. By default in the docking step, the binding site doesn't move too much. It will have some slight movements just to prevent overfitting, but you can release the whole binding site, and then during each docking step, it'll then minimize the binding site and residues around it. This obviously takes slightly longer, but it's still pretty reasonable. Also, in the post-processing step, if you want to hold the protein still, you can. I don't think for the protein you can specify individual residues yet that you would like to move or hold. It's mainly just the atoms within the ligand.
When you were demonstrating the system ligand approach, and you were showing the centroids for the ligands, how does one actually obtain those coordinates?
Oh yeah, sorry. Maybe I brushed past it. It's with the alpha mask, the output of that, you get what you saw. I'll go back to it here. Is all of these densities, and you'll get a combined map with all of them. You'll also get individual maps of each one, and with an ID, and a JSON file with the ID and the centroid of that segmented density. And you just use that for the centroid. Alternatively, you could use something like Chimera, just pick a few atoms within the binding site or around the binding site, calculate a centroid there and use that. The centroids don't necessarily have to be too accurate. ChemEM will do a lot of work in the background to find a binding site and make sure what you've put in is sensible. So you don't have to be like bang on. I guess you kind of do if you want to segregate the densities, if it accidentally is pulled two into one. But generally, it's okay with just a loose approximation of where it is.
What is the lowest resolution map one can effectively use? And to add a little bit to that, as one uses lower and lower resolutions, do the stereochemical restraints get weighted more and more heavily?
I mean, we haven't really tested TEMPy-ReFF below 10 angstroms. I think you do have to, of course, restrain your system, and you can use the rib-fine restraints to maintain the rigid bodies. Of course, I think with nucleic acids, you might be able to go a little lower because often the RNA helices are a little bit more visible in the density. But yeah, I can't actually tell you a sort of a lower bound there, unfortunately.
For ChemEM, I probably wouldn't go lower than 6. Just at this resolution for such small molecules, you haven't really got the fine detail to make a credible model anyway. It will still do it. But I guess how you interpret that now is, I'd interpret it with caution, definitely.
So you've spoken about fitting into EM maps. Do these methods work for crystallographic maps?
No, not currently. I guess crystallography have their own kind of software which works great.
Yeah, TEMPy-ReFF's not designed to work with crystallographic data.
A couple of questions about searches for ligand sites. One, is it possible to dock a ligand that may have density for multiple conformations of that ligand?
Yeah. Well, I have experienced this before with a small molecule that had multiple conformations, and you could there was clearly one preferred conformation and then one that was, I guess, the minor conformation, which had a benzene ring that had moved. And when you dock stuff, the output you'll get essentially, it will pick how many iterations it needs to go. And for each iteration, you'll get a solution, and then it will rank these solutions. And usually the correct one is within like the top 10% of solutions. And what I found with this situation was that a lot of the higher up solutions I got were for the preferred conformation. And as I started to move down and down the list, there were some that were trying to fit to this kind of alternate conformation. Another thing we did was, but it was more of the map. We had a ligand that had multiple conformations, but in this approach, we went back to the map and tried to tease out both of the conformations and then fit. So I guess that didn't.
Yeah, that's the serotonin receptor, right? Yeah, there's a preprint out there which explores that.
But yeah, so yeah, it's possible, and you may or may not get both conformations within the solutions given back. But however, I only did it for one case, so I'm not sure if it's applicable to all cases.
And to follow up, is it possible to force the searches to look for multiple conformations?
It will kind of do it naturally anyway. With the docking, that's what it does. So how the docking works, I didn't really have time to go into it here. Is essentially it'll break it down into a grid of what's possible for where your ligand can be, and start kind of searching all of these different positions initially randomly, and then as it kind of learns more about where this thing likes to go, they should converge and converge into areas that it likes to be more. During this phase, and all of these solutions are given back as where it's searching. And during this phase, it's possible for it to move into this alternate conformation. I guess if you wanted to kind of fudge it, you could add in an atom or two as an SDF file for where you didn't want it to be. I have never tried this and wouldn't, and then it won't move into that one. But yeah, I haven't tried it. I wouldn't really advise it. But maybe it could give you a starting point that you could then refine something new. But unless I guess you can tease out the two conformations, I'd actually recommend going back to the maps and trying to get out both conformations.
And for these searches for multiple conformations, what is the minimum map resolution you might think you need to effectively find them?
I have no idea of a concrete answer for this question. So the one case I came across it where I did get both conformations. I think the resolution was about just over 3, and it was a pretty good just over 3 within the binding site. The resolution where we went back to the maps and tried to do a conformer search kind of in a 2D and tease out both conformations was about 4.
Yeah, I think it was
about 4, but it wasn't a good 4 around the binding site, especially. It wasn't really a complete binding site. So yeah, I'm sorry, I don't have a concrete answer. You just kind of have to try, and it's pretty quick to run ChemEM anyway. It could give you an answer.
All right, to switch gears a little bit, is it possible to use these methods on just nucleic acids and nucleic acid protein complexes?
Yeah, I mean, TEMPy-ReFF will happily refine RNA as well as nucleic acid complexes. This is totally possible.
ChemEM is more meant for just small molecules rather than whole complexes.
So I'll throw in a personal question. You mentioned early on that one can incorporate information from other techniques. How is this done effectively? So let's say one has NMR data, maybe NOEs are giving some kind of distance restraints, or RDCs are giving some kind of angular restraints. Are those imported in just as harmonic restraints, or is it something more sophisticated than that?
Oh, okay. This probably is going back to the sort of introduction slide. So, I mean, in the lab, we work on integrative methods, but the two methods we just discussed are really aimed just at CryoEM data. But certainly adding in additional restraints is something that we're very interested in. So these could be harmonic restraints or some other kind of positional or relative position restraint. But it's not something we currently support in TEMPy-ReFF or ChemEM.
All right. I think that brings us to the end of the Q&A. If no one has any additional questions and Justin is ready to get started a little bit early, we can do that.
Yeah, sounds good.
Otherwise, thank you, Aaron and Tom, both for a really great talk.
Thank you.
Thank you.
Cheers.